need a booster seat in my chair. The height of this camera and everything, I have to put my leg underneath my leg.
Oh, no.
So that I'm at a good height. Otherwise, I'm like...
My laptop is on top of a moving box and a chess set.
I need a booster seat.
In order for it to be the right height.
I need an Apple box booster seat. Right height, wrong height. We got it. We got it. We're here. We are live. Hello, everybody. I hope you're out there. Are you out there? We're here. We're here ready for science. Blair's sick. She has a creeping camp crud.
Yes. That's the official scientific name.
It's better than the cruddy camp creep.
Yes. Unequivocally, I would say.
Yeah, there's that. Oh, hello, everybody in our YouTube chat room. Hello, everyone in our chat room. We're here. We're ready to start.
Hello, everybody not in the chat room.
Hello, everybody not in the chat room. Who's watching this later? Not live. Are you ready for a show? I'm ready for a show. Let's do a show. Let's make a show happen, okay? I think we can do this. Yes. Is everyone ready? Are we agreed? It is time. Yes. TWIS live chat room. Are we doing well? We're ready to go? All right. Fada says we look and sound great. Ed from Connecticut says everything looking and sounding great. Awesome. Starting in 3, 2. This is TWIS. This Week in Science, episode number 680, recorded on Wednesday, July 25th, 2018. Sneaky science. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with spiders, sneakers, and beer. But first...
Disclaimer. Despite any evidence you may have heard to the contrary, though hopefully you have heard none, you are one of the most important Earthlings in the history of planet Earth. Yes, there have been a lot of Earthlings throughout history. For me to compare you to all of them in order to come to this conclusion may seem daunting. And yes, it is possible that my lack of direct knowledge of who you are makes this determination dubious at worst. Yet I stand by the assessment because the following is certainly true. 1, we live in an age where idle-minded activities are easily available everywhere. 2, we live in an age where time is money. The baby needs a new iPhone and the rent was due yesterday. 3, we live in an age where people can choose how educated they want to be or how ignorant they would prefer to remain. And 4, you have passed on idle-mindedness to take valuable time out of your day to add to your knowledge, which leads us to 5 people who do that are always the most important Earthlings the planet has ever produced. Because 6, if not for Earthlings like you, if we should ever lose those with the desire to seek knowledge, the Earth that would follow in your absence would be such a thing of horror as to plunge a planet into darkness and despair, a living nightmare of endless lies, endless arguments made with great confidence, yet utter ignorance, endless propaganda, endless war, George Orwell's vision of a boot stomping on a human face forever. That's the world without you, and why you are one of the most important humans ever to breathe the Earthling atmosphere. So thank you. Keep up the good work. And as a reward for your efforts, here's another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin and Blair and everyone else out there. Welcome to another episode of This Week in Science. we are back yet again to talk about all the science that we enjoyed finding in the last week, bringing it to you, curating the world of science news for your ear holes. And if you're watching online right now, for your eye holes as well, maybe. I don't know. But here comes the science. We're coming right at you. I brought stories about water everywhere, a battle, biological battle that's taking place. And what was the last thing I had? Oh, yeah, the fungus among us.
Ooh.
Justin, what do you have?
I've got chatty bacteria, better beer through science, a never-aging brain, and sex, lies, and survey results.
Oh, boy. We're going to save that one for the end of the show, I think. Tease it now, everyone. And Blair, what's in the animal corner?
Oh my goodness. I brought swelling spiders. I brought pilfering primates and the Endangered Species Act under attack.
The Endangered Species Act.
That's right.
It is so true, and it's really sad.
We're going to talk about it.
You guys. Okay, I can't wait to talk about it. Let's talk about it. Let's talk about it. But first, as we get into all of this, I would like to remind everyone that if you are not yet subscribed to the TWIS podcast, you can do so on YouTube. Also, go to twis.org where you can find the wonderful links for subscribing. We are available in all of the wonderful places you find podcasts. On to the stories. Let's start off. You guys want to start off with water, or do you want to start off with a biological battle?
I think water was the start of it all anyway, right?
Okay, so let's talk about this water everywhere. And we are finding it places potentially, not necessarily, but potentially, researchers looking at Mars have detected a sizable aquifer.
An aquifer, you say?
Yes, a sizable aquifer under the surface of Mars. So aquifers, we know we use groundwater. This is an aquifer. It's a store of water held within the rocks under the earth. It's water that doesn't absorb into the mineral materials of the crust of the earth. And so it's held there in pockets. And it's wonderful. It's great. It allows us to survive. It also could potentially be a source for underground life. Yeah. All right. So between May 12th and May 2012, and December 2015, Roberto Orosei and his colleagues used MARSIS. MARSIS is the Mars Advanced Radar for Subsurface and Ionosphere Sounding instrument that's on the Mars Express spacecraft to look at a part of Mars that's called Planum Austral. It's on the southern ice cap of Mars. They looked at 29 sets of radar samplings, mapping out an area that had a really distinctive change in its signal, about 1.5 kilometers underneath the surface of the ice on the southern pole of Mars, and extending outward about 20 kilometers. There's this profile that is very similar to lakes of water that have been found beneath the Antarctic and Greenland ice sheets here on Earth. And so we use a lot of what we see here on Earth to infer what could possibly be going on other planets, given what we understand about physics and geochemistry and geology. And so this is a suggestion that there is this subglacial lake at the South Pole of Mars. So the temperature is expected to be below the freezing point of pure water. Orosei and his colleagues in the paper, they do note that there may be dissolved salts that are known to be in the rocks on Mars, like magnesium, calcium, sodium. Anyone who's ever made a solution, a salty solution, and tried to freeze it, it can get colder before it actually freezes. Salts lower the freezing point of water. So could be briny, a salty underground aquifer under the ice that's not frozen. So the possibility is it's briny, it's salty, it's potentially watery. We don't actually know if it's water because this is just radar, but based on what it looks like, could be.
It's not like radar's a new thing. We've had quite a bit of practice with this radar technology. If it looks like water because it sounds like water, it's probably water. So how do we get a submersible rovery robot
How do we get
to go
it
take a look?
Crash into the South Pole of Mars? So one of the researchers says something, it could be other compounds, other liquids, other things are liquid it's not just water necessarily, but Roberto Orosei, he says to The Verge, he says, I've run out of ideas on how to explain this in a way that isn't water. We've tried to exhaust every possible alternative, and we think we've done it. So they think they have absolutely exhausted all alternatives and that this is water. Liquid lake of water. Yeah, and if it's salty, dude, lifey.
Mhm.
That's where life is on Mars.
It's probably lifey.
It's probably lifey.
Yeah.
Right.
It's also probably a good spot to open a spa. Just float in that salty water.
And freeze.
It's good for you.
Let's send a robot.
It gives another meaning to those polar bears.
We'll send Blair
for the
and a robot.
plunge.
Yeah, I'll go. I'll tell you if it's water or not.
Yeah. But the question is, if there's one, are there more? Will further radar sampling and mapping under the surface of Mars find more pockets? And, the North Pole and the South Pole, these are the colder areas, colder regions of Mars. So, maybe something mid-latitude, maybe it wouldn't be so freezy. Maybe that, if they found water in other places, maybe that would be an even more habitable spot on Mars. Yeah.
So Fada in the chat room brings up an interesting question. How do you know it's water and not another liquid?
Right. So that is a really good question.
Well, the likelihood
They
that
know.
it's-
It's a science
likelihood-
thing. They know it. Right.
Well, the likelihood that it's beer or wine is probably more far.
Well, beer and wine are still mostly water. So
So the researcher, it's radar, and the signal is similar to radar signals that are water here on Earth.
because it's things like density and stuff like that, right?
Yeah, there are other measures that go into it. So they can. They're hoping that there's a new lander called the Mars Insight, which is planning to probe the interior of Mars to figure out its internal temperature. So that could help determine whether liquid water is possible. And once we know the temperature of Mars, that will help to determine whether or not even a briny water solution could possibly remain unfrozen under the surface of the South Pole. Yeah,
submersible, like ground penetrating robot with a viral and bacterial detector. That's what we need.
And to not bring any of our own, which is pretty much impossible.
Secondary consideration.
Yeah. Well, because that's the other thing is if it really is water, if it's briny water, and we send anything into it, there will be life there, whether there was before or not.
And there'll be life on 2 planets for sure, which will be awesome.
Yeah.
Totally worth it.
And that's it. I'm sure it's only the 2.
And we can find that there was life there, but that we devastated it upon discovering it, which is too bad. But now we know, right? Which is half of why.
See, how the heck do you do this?
I don't know.
Okay, from the abstract, before I jump to the next story, from Science from the article in Science where it was published this week. It is the dielectric permittivity that they used to determine that it matches water-bearing materials here on Earth. So the physics of a dielectric substance
Wow.
are what they were looking at, and that they were able to determine from the radar signal.
Huh.
Yes. So that's what you're going to have to go look up, everyone. Go look up dielectric permittivity. It's your homework for this week.
It's dense. I'm already looking at it. It's a bit dense. There's lots of graphs and numbers involved. And constants I've never heard of.
Although my new favorite word is radargram.
Oh.
I'm going to send you a birthday radargram. Yes. No. Moving on up to another story, biological battles. Thanks to water, life has occurred on this planet. And rocks, South America, they have determined they're 3.22 billion years old, and thus the location of the oldest life on land on the planet. 3.22 billion years. That's what they've got. They've got it. But anyway, moving into that life that has been evolving over the past 3.22 billion years, bacteria, it's where it got, they're one of the first, and viruses right along with them. And we've talked a lot about bacteria and the viruses that attack them, the phages. Something we have been very interested in is possibly using phages as treatments for antibiotics, for bacterial infections that are antibiotic resistant. So,
which in the past, the ways that this has been attempted were huge failures. But we have to keep in mind, when these were tried in the past, people were just drinking concoctions that were full of viruses without really knowing
they're trying to match them up. Yeah.
But this is in the way, like, this has a bad rap in some scientific circles. But if you look into the details of how this was first attempted, it was not done sanely.
Which is unfortunate. I mean, let's try and approach things from a sane direction, which is where this study, or there's a couple of studies that are just out that I think they do approach it from a sane perspective. They're looking at the biology, the ecology of the bacteria-phage relationship. Bacteria create the CRISPR-Cas9 system, which we currently use for gene editing. We went, hey, look at the bacteria using this neat system of molecules to snip up DNA and protect itself from viruses. Let's use it ourselves to snip up our DNA and mix things up a little bit and do some gene editing. So we took advantage of a biological system that's already doing very great things for living organisms for our own purposes. Now, getting back to the CRISPR-Cas9 system and what it does for bacteria. So this CRISPR-Cas system, it snips up viral DNA and makes it so that the viruses, these phages, cannot attack them anymore. But the new papers that have just come out about the battle that's waging, the evolutionary pressures that are on these systems to change and adapt are super strong. So these researchers at the University of Exeter have determined that phages, according to the lead or one of the co-authors of one of the papers, shows that phages can work together to disable bacterial immune systems. And it has important implications for using phages to treat human infections, since the dose of phage that is used can determine whether the phage is able to kill the bacteria. So what these two papers together have found is that sometimes it's just one group of phages. They come in, they attack the bacteria. The bacteria attack back with CRISPR-Cas9. And if there's enough of the phages producing a particular protein, a molecule to combat the CRISPR-Cas system that's called now, so originally by the researchers, anti-CRISPR proteins, otherwise known as ACR proteins. These ACR proteins break down the CRISPR-Cas9 system so that the bacteria no longer can defend themselves. And then the phages take over. If there aren't enough phages not making enough of the protein, they can't take over. The bacteria win. However, a first wave of attack, the phages producing even a small amount of this ACR protein, it can do damage to the bacterial defenses so that they're not strong anymore. It's just like, weakening the defenses for the second wave of attack. So researchers are thinking that there may be, because of this dose effect of the anti-CRISPR proteins of the viruses, that there's almost like an altruistic effect of the first viruses that go in sacrifice themselves for the good of the viruses that come later, because once the bacteria are weakened, the viruses will prevail. Yeah. So it's this amazing battle system that is ongoing at a very small level around us constantly. But understanding how it works, we could potentially make use of it to be able to help ourselves heal from infections.
Yeah. And as we've discussed many times in the show, it is sort of the underpinnings of our initial immune system. We start not with a healthy, thriving gut microbial system. We start with a really heavy viral load that eventually trims down our bacterial load to that which is right for the human gut. So that's why, I mean, it seems like these should be the viruses that we're using as phages. We should just go back and steal viruses from infants or borrow. I don't know how you
steal.
I don't know how you do that.
You're not taking candy from babies. You're taking their viruses.
I'm taking a virus load that you need for your immune system. Well, so I don't know how you do it, but it seems like these are the viruses you would want to sort of focus on for doing these sorts of things that called your immune system and your gut microbial system its natural habitat at some point.
Absolutely.
Yeah. I've always loved this idea. This has always seemed like this is the way nature does antibiotics. You know, we should always try to follow nature's lead.
I think antibiotics are, natural, have been naturally sourced for years. We usually do find the molecules that we use for antibiotics, the different families of compounds, have come from watching bacteria attack each other, taking the compounds that seem to work and that bacteria use to fight each other. That has been what we've used. But at the same time, they've only lasted so far, and we need other methods. And this phage method might be another way in there. Use the phages, figure out exactly how this system would work. I don't know. Here, we'll give you a round of antibiotics. That doesn't work. We're going to throw in a few viruses as well. What? Viruses? No, not viruses for you. Viruses for the bacteria. We're going to make the bacteria sick. Yes, I'm in.
But if we really, if this is our planned counterattack, we should probably stop talking about it.
Why? Why should we talk? What?
Because if the bacteria catch wind of it, they may tell other bacteria. Because it turns out microbiologists at the University of California, San Diego can now explain how communities of bacteria talk to each other.
What?
They can effectively relay signals across what would seem to them, at least from their perspective, to be a really long distance. So this is biofilms is what they're looking at here. These are things that were once considered to be just sort of clumps or clusters of microorganisms. They've been seen utilizing ion channels for electrochemical communication amongst themselves. So we've already seen, some sort of in biofilms in the past that we've talked about, that they can have differentiation in terms of compounds that they produce. So they sort of start to individual bacterias start to take on different roles within a community. Like you actually have ones that are manufacturing this substance versus those that are just gathering nutrients on the outside edges.
Right.
But really no explanation of how this differentiation of activities comes about. And it may just be that they're group thinking, if you will. These are findings by Joseph Larkin, senior author, and senior author Guru Sewell of UC San Diego. They've published this just today, the 25th of July, in the Journal of Cell Systems. So these communities of cells, these are all around us. These are in soil. You can find it in sewers. You can find it on every sort of surface, and even on, Blair, the surface of your teeth. These are...
Go brush your teeth.
And cells at the edge of a biofilm community tend to grow more robustly than the interior ones, because they're on the outside. They've got more access to nutrients, and to keep the edge growth in check and ensure that the entire community stays healthy. And the hungrier interior members of the biofilm interior send electrochemical signals to members of the exterior saying, "Send us food," or something along these lines. And these signals halt consumption on those edge bacteria and they start passing nutrients to the interior so that the interior does not starve. And also, this also rings back. We reported about this, talked about that effect in the past. Carl Zimmer, New York Times, was on the show. He was talking. It was at the time we were dubbing it an altruistic thing, where if these colonies or these biofilms are in a sort of famine state, the outside edge ones will pass nutrients to the interior, even if they themselves are starving to death and dying. So these signals from the interior to the exterior are important enough that the cells in the exterior will ignore basic biological, instinctual, built-in programming for self-preservation and will feed the interior. And now we can see basically that there's a signal that is coming to them saying to do this. Quotey voice of Joseph Larkin, UC San Diego. So feeding about the allowing nutrients to pass to the interior. This keeps the interior fed well enough. And if a chemical attack comes and takes out some of the exterior cells, then the protected interior is able to continue and the whole population can survive. It is essential that the electrochemical signal be consistently transmitted all the way to the biofilm edge, because that is the place where the growth must be stopped for the community to reap the most benefit from this signaling. And approaching their new study, researchers sought to explain how bacterial communities are able to propagate these electrochemical communication signals. So it sounds very brain-like, right? Ion channels and like, but they don't have, of course, neurons. There's not these pipelines of conductivity of sending ions up and down between the cells. So they have instead structures to relay these electrochemical signals known as axons. These are bacterial communities, or that's what the neurons have. So they were trying to figure out how are they doing it. So they went through all kinds of data. They began collaborating with Purdue University. This is Andrew Mugler and Xiaoling Zhai who proposed the idea that percolation theory could explain how bacterial communities may be propagating a signal. Percolation theory has been, I guess, around since the 50s. This is how coffee is apparently made.
Yes,
hot water,
percolating coffee maker. Yes.
Hot water goes through the individual coffee grounds, and then in the oil industry, they sort of create pressures to percolate petroleum out of the sand. So in the community of bacteria, the signal passes from cell to cell in a connected path over a distance of hundreds of cells. And then using a fluorescence microscope, the researchers were able to track individual cells that were firing at any given time or transmitting the signal. They found that the fraction of firing cells and their distribution in space precisely matched their theoretical predictions of the onset of percolation. In other words, bacterial community had a fraction of firing cells that was precisely at the tipping point between having no connectivity and full connectivity among cells, also known as critical phase transition points. So, what does this mean? They were effectively able to transmit this signal. It sounds like they're using percolation. It sounds sort of like a community sourcing sort of thing.
But they have these cell to cell connections and these pores that connect each other. And so they are, even though they don't have specialized electrochemical transmission channels, just like nerve cells do, it's amazing that they're able to propagate a signal that certain cells go, I need to send a message, and that message gets sent by the appropriate cells that will get the message to the outside.
Yeah,
and to get the information in. Yeah,
and I wonder if it's also like a little bit of just sort of like, it's not who wants it, but who's stuck with it. Because at the end of it, like you're like, okay, here, pass this on. Okay, here, pass this on. Okay, here, pass. Yeah. Oh, there's nobody there. Oh, I guess it's for me. I'm the one. I'm stuck with the message because I have nobody to give it to over here. So now I have to give them nutrients back that way. This is not a good deal.
Stop taking the nutrients and hoarding them for myself. Give them back. Okay. Yeah. Pass it back. Pass it back. Pass it back.
I mean, that's the amazing thing. Like that's actually to me the more amazing thing about this discovery isn't just that the signal is being sent, but that it's built in to elicit a specific type of response. That the message is not just a signal is being sent, but that it's being received, understood, and acted upon. That's the really wow.
And these are, like you said, these are single-celled organisms. They're very simple. We've talked before about slime molds and their ability to quote unquote think, to move and act as these fairly intelligent organisms, which is, I'm sure many sci-fi aliens have been developed based on slime molds, but now just bacterial biofilms in general. And it's fascinating to think that, the biofilms in my bathroom or the biofilms on my teeth, they're communicating within themselves.
And so we already know there's there's gut brain communication that takes place. Could it actually be as simple as they have signals? They've got like a registrar of, and you know what? It's not going to be the most verbose communications, but it could just be,
sugar.
Enough.
No more sugar.
More sugar. Why do they keep sending more sugar?
The thing is, this is when I hear it though, it sounds a lot less complicated than things, for example, that the slime molds do, just because it is, since they respond to it whether or not they have enough nutrients for themselves on the outside. That means it really is, it more sounds akin to me to what we did at Sketchfest at the Cal Academy with the electrical signals in Justin's arm, where you're receiving it. Exactly, it's involuntary. You're receiving a signal, electrical signal in that case, and there's an output from it. And in this case, the output is to send nutrients. And so they're doing it whether or not they have enough for themselves, which means it's not he says he needs more nutrients, let me send along some nutrients.
It could be that, or you could be massively underestimating the sacrifice and altruistic
the altruistic nature.
Absolutely.
Bacteria.
Edge bacteria in a microfilm. That's a noble position to hold within that.
Yes. Oh no, you take it. You need it more than I.
Oh my goodness. All right. Well, that does it for me. I've had enough. I can't take it anymore. Because it's time. It's time now for Blair's Animal Corner.
Loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
got, Blair?
Well, let me tell you, I had some stories in the rundown today. I put something in the short stories for later. But since I did that, I've decided that this is very important and I want to talk about it here instead, kind of more long form. And that is the news about the Endangered Species Act. Since, you know, we won't have a Blair's Animal Corner if there aren't animals. So
this is very selfish.
It's kind of an important thing to talk about. So I'm not going to get into the politics of it. I will suffice it to say that there are recent attacks on this act. That isn't new. More on that in a minute. But I wanted to talk about research on how substantial the act is and how well supported it is. Because if you hear the people who are attempting to restrict it and you hear the people who are opposing it, it sounds like a very divided issue, much like other things that we've talked about on this show, climate change, evolution. You know, there's just scientists are divided on this. No, they're not, which is what this is about. So this is a study asking, quote unquote, ordinary Americans about the Endangered Species Act and how they felt about it. This is from Ohio State University. And this is a survey of 1,287 Americans from many different walks of life and many different interests.
Are they all in Ohio?
They were not all in Ohio. And they found that overall, the numbers were that 4 out of 5 Americans support the act and only 1 out of 10 oppose it. So the math there is that 8 out of 10 are in support of it, 1 doesn't really care, and 1 actively opposes it, right? This is, you know, 4 out of 5 dentists recommend Trident. 4 out of 5 Americans support the Endangered Species Act, right? So it's good enough for your gum. It's definitely good enough for the Endangered Species Act. These numbers have remained stable for 2 decades throughout surveys. And what they found was, digging deeper into this data, on average, approximately 83%, that's that 4 out of 5 of the public support it. So it's definitely not, quote unquote, controversial. When they looked at interest groups, 8 specific interest groups, including hunters and property rights advocates, they were all at least 68% supportive, which for politics is still pretty supportive of anything. And that was consistent throughout regions of the United States. It was not geographically linked at all. About 74% of conservatives, again, very high, 77% of moderates, and 90% of liberals supported the act. The highest...
Who's the 10% of liberals who are against it?
I don't know. That's a good question. The highest percentage of active opposition to the act was found from property rights advocate groups, which makes sense, right? You don't want to halt construction for that endangered owl or that salt marsh harvest mouse, as is happening in the East Bay of California right now. 21% of property rights advocates said they were against it. So that's still not very much. But now this all leads to where we are today. So over the past 2 decades, pretty much across the board, the lowest kind of consensus to keeping the act was 68% out of any one of these groups. But when you look at attempts to attack, amend, repeal, curtail the Endangered Species Act, there were roughly, from the 1990s and the 2000s, there were roughly 5 attempts a year to adjust, curtail, remove parts of the Endangered Species Act. 5. From 2011 to 2015, there were 33 attacks per year. In the past 2 years, guess how many?
110.
I'm looking at the number and it's just making me hurt inside.
150. 150 attempts to attack, amend, curtail the Endangered Species Act in the past 2 years. And one of the lead researchers...
Yeah, no doubt.
Yeah, right. One of the lead researchers, Jeremy Bruscotter of Ohio State University, he says, I don't think at any time, maybe since the act was passed, have there been this many members of Congress working in direct opposition to the act. But that doesn't mean that they're acting in the interests of the people they represent. The co-author of the study, John Busetich of Michigan Technological University, says the Endangered Species Act is a genuine point of American pride. It's considered to be one of the best laws in the world for conserving biodiversity. So why don't our representatives represent us?
That is an amazing question. Why not? Why?
Yeah.
Why are the representatives not representing the people? Because I'm not going to talk about politics.
So
I'm
this
gonna
is
say
this
the
is
things
my
that are in my mind. Yes.
This is my non-political pro-biodiversity statement that I am going to say right now. And the reason that I talked about this in the main part of the show is that this is a good reminder to all of us to let our representatives know what is important to us, whatever that may be. I'm not telling you to vote for a specific person or to support a specific thing, but if biodiversity is something you care about and the Endangered Species Act is something you care about, we now know that our representatives are not mirroring our opinions. And so this is the moment to do something about it when attacks on that act are at an all-time high by a crazy number. So there we have it.
Yeah. We need, yes, more voicing of opinions to our representatives.
Yes.
Yes. And everyone out there, I know college students are going to be heading out to colleges around the country very soon. Many many students just heading out for the first time, maybe who are just going to be 18 and voting for the first time as well. Make sure you understand whether or not you are registered to vote. Find out if you're registered by, getting a driver's license in that state, or if you need to actually send in a registration or register online, whatever it happens to be. If you're a college student and you have not done that yet, it behooves you to have a voice and to be able to vote in the midterm elections that are coming up. Additionally, several states are starting to purge their voter registrations more regularly, and people are finding themselves suddenly unregistered. So maybe if you haven't checked in a while, check and see if you're registered.
And while you're at it, register to vote by mail because it is the easiest. And it means that even if you have something important going on that day, or you feel like you can't handle the pressure of the booth when you're trying to decide what to vote for, you can do it all ahead of time. I love it. I can do my homework, have my laptop out, multiple reference pages, make my decisions, jot it all down.
As long as you mail it well ahead of voting day, because those mail-in ballots do need to be received before or by the vote day of voting. So there we go. This is our public service messaging right
there's
now.
A little bit more. To the optimistic, right? Pessimist. Or no, to the optimistic procrastinator who went to the point of getting a mail-in ballot, but then just totally put off filling it out and sending it in. Yeah. You can actually still drop it off at the polling place on the day.
That's what I do. That's what I do every time.
I've done that a few times.
And I can still get my sticker.
Exactly.
Sticker.
So let's quickly move on to a nice, fun story that actually might give us a glimmer of hope before we go into the break. And it's all about
hope. I have hope. People are going to go vote, and I have hope.
Yes, that's right. Well, you know who else can give us some hope? Wolf spiders.
Oh, wolfies.
Wolf spiders. Wolf spiders in the Arctic. Did you know that wolf spiders are so abundant that they outweigh real wolves in the Alaskan Arctic by several orders of magnitude?
Wow. Spiders.
Wow.
I just got goosebumps all up my arms.
The good news is, according to Washington University in St. Louis, that weight might be increasing. And that's actually good news for us. Let me explain.
And that weight, it's more spiders?
It's bigger spiders. So in a warmer future, as we know we're heading towards, that good old 97% that we talked about before of consensus of climate change and all that good stuff. in a warming world, spiders and other arthropods will be bigger. We know this, right? And in the Arctic, this is also true. The wolf spiders are going to be larger and more prolific. Why is this good news? Well,
That's rad.
it has to do with what wolf spiders eat. They don't make webs. They don't eat flies. They actually hunt on the ground. They can eat almost anything smaller than them. And their favorite food is the columbola, which is a small arthropod. It's called a springtail, usually. And the spiders eat these guys, but springtails eat decaying plants and fungus. And in the wet tundra, fungus controls how quickly dead plant matter decomposes. Stay with me.
You know what?
When the spiders eat springtails, the springtails eat fungus, right? So then there's more fungus when there's less springtails. Fungus grows unchecked. When there's more fungus, there's faster decomposition. But faster decomposition causes a problem because it releases carbon dioxide and methane, which increases global warming. The good news here is that large wolf spiders will likely eat things besides springtails because the wolf spiders will be too big to eat them. And they did figure this out in an actual experiment. They set up experimental enclosures in the Arctic tundra of northern Alaska over 2 summer seasons. They had, in these little mini ecosystems, they had some animals, they had some fungus, they had some spiders. And what they found was that they changed the temperatures in the different ecosystems. What they found was that at ambient temperatures, at normal current temperatures, there were fewer springtails in the high spider density plots. Decomposition of leaf litter was increasing, therefore more greenhouse gases, right? But in the warmer plots, the researchers found more springtails, less decomposition, less fungus. So what it looks like here is that bigger spiders reduce the rate of decomposition, and so it will reduce the runaway effect of warming as ice melts and things decompose.
That's fascinating. I wonder if that would just work everywhere. If we should just start spider breeding programs.
Nope.
Nope.
No.
Nope.
We should just have more spiders, and then we would have less warming because it's been lack of spiders this whole time. It's been.
I'm okay keeping the large wolf spiders in Alaska away from me. It's interesting because this is a study, instead of looking at direct predator-prey relationships and how they're affected by a change in temperature and a change in environment, this is a biome level change, right? So this is a carbon cycle level change, right? This is a huge big picture change that's happening, which more than anything is a reminder to us that as the planet warms and things adapt and things change and bugs and spiders grow bigger, there are going to be these cascading effects on our ecosystems that we can't even imagine yet. And so studies like this are super important so we can get ahead of these things and we can predict what's going to happen and help kind of get ready for the curve. But it's also important to kind of take a step back and remember how deeply rooted and connected everything is. To think that a wolf spider could affect methane gas release from the melting of permafrost seems insane until you connect all these dots. It's very interesting.
Yeah, it will affect it, but the actual, the how much, what percent of the overall change is it affecting? That is a big question. And my other question is, if these animals are not, these insects, spiders, if they're not eating springtails, what are they eating?
Spiders.
And they're eating other spiders?
Yeah.
But that in itself could pose a problem. So
indeed it could.
But then you also are overlooking the fact that you'll have much fatter birds. from all the
There's
available
that
spider
too.
spiders.
Which means
As
more
long as
poop, which means more meth. Oh no.
Yeah, here we go.
Yeah. Well, the
Oh.
bird poop is at least more nitrogenous.
Yeah, there you go.
Mhm.
Sure, that's the silver lining, I guess, or the
Yes.
whitish greenish lining.
Yeah. No, it's very, it's an interesting question. If these spiders do get bigger and if other spiders also get bigger with the warming climate, how will that change what their prey species are? How will it change the dynamics of ecosystems, especially when we consider the massive amount of biomass that spiders and other insects entail on this planet?
Yeah, remember, spiders on this planet eat more than all of the whales on planet Earth combined.
I know. And yeah, this is mind-boggling.
Yeah,
Kiki,
we
Kiki,
talked
do you
about
have-
it on the show.
I didn't realize this. I've missed this all of these years. Kiki, do you have a problem with spiders? You seem to be a little.
No, I'm
Hello,
cool with them.
I'm talking about spiders, and
I'm
I
cool.
think I've
I'm
noticed.
cool with spiders.
I'm not sure you are.
It sounds like she just doesn't want them near her face, which is how I feel about them as well.
But that doesn't like
Yeah.
do you have that react? I would have that same reaction about just about every animal, no matter how cute it is. Like I still don't want
Wow.
it on my face. Like why would I want
You don't often kind of like walk through a door early in the morning and go, huh, there's a cheetah right there.
No,
You don't
but
wake up to a bullfrog jumping across your face. You know?
Not so often.
No.
Doesn't
No,
come up
not
very
so
often.
No.
No.
It's a uniquely arachnoidal issue.
It is. That it is. All right, everybody. Have we come to the end of the animal corner from now?
We sure have.
Okay, we have everyone. We are going to be moving on forward. This is the end of the first half of This Week in Science. We'll be back after this short break with more great science. There is Fungus Among Us to come. We've got sneaky mangabees, little sneakers. They're on the path, and much, much more coming up. Oh, yeah, beer. There's beer in the second half of the show. You don't want to miss that. We'll see you there. So stay tuned for more This Week in Science.
Can't explain things you've heard with more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
Oh, have you plotted the data? Have you scatter plotted it? Have you looked to see where the answers lie? Where should you go? Where should we be? You're in the right place. This is This Week in Science. I hope you know that if you're just catching it right now. I hope you do realize what you're listening to. Thank you for joining us once again for another episode of this show. I would love to talk with you a little bit about how we keep this show going and how you can help out. I mean, aside other than, yeah, there's lots of connecting words there. Other than just viewing or listening to the show, you can help TWIS out in a number of ways. TWIS relies on viewer support to maintain its production schedule. We try to produce shows almost every week of the year. Wednesday nights, 8 p.m. Pacific time, we're online and we are bringing you science news. We love to do it, but there are some basic costs that we need to address to be able to have the technology and to bring the show to you, and also make use of our time. Absolutely, to get it here. So all of you out there, I would love you to head over to TWIS.org. TWIS.org, if you have not been there before, is the main portal for the TWIS universe. If you head there, you'll see a big yellow orangey subscribe button. Click on that. It will give you links to be able to subscribe to us on YouTube, iTunes, and Google Play. Hey, I saw today that we're well within 100 subscribers to get to 12,000 subscribers on YouTube. So if you're not subscribed to us on YouTube, please do that. Yeah, there was something we needed something like 89 subscribers this morning. You can be one of those. So subscribe to us. Click on that YouTube link and subscribe. We also are available on iTunes, Google Play, and other outlets, but iTunes and Google Play and YouTube, you can subscribe easily to right from twist.org. So you don't have to remember all sorts of stuff. Now, beyond just subscribing helps us out a lot so that we know how many people are listening to or watching the show. But if you can help us out in a financial fashion, one way you can do that is to head over to our Zazzle store. So click on that Zazzle link at twist.org and find all of the Twisty goodness that is over at our store. We have mouse pads and shirts and cups and hats and all sorts of wonderful things, many of them with just the Twist logo on them. However, others are sporting the wonderful original art from many Blair's Animal Corners from past years. We've got tortoises and nautilus mouse pads. There's a mammoth lumbar pillow. I think that is one of the most fabulous items we've got on there. But that's just me. You'll find your own sciencey, twisty goodness if you go peruse the store. A portion of the proceeds do go to support this show. Also, heading back to twist.org, you can scroll down the side and click on the yellow donate button, which will take you to a PayPal interface, which you can use to donate one time. If you just want to donate one time, you can click on that donate button. And that's what you'll get to do. If you want to donate multiple times, like say every month, but you don't want to have to remember to come back and you're like, oh, maybe I could have like a recurring cost on my credit card or my PayPal account. We have an answer for you. Go to any of the most recent episodes. You can listen to the episode right on the show page, the episode page, or you can also find the show notes that are available there with links to the stories that we discussed. And at the very bottom of the page, along with a place where you can make comments and talk with other people here, pink buttons. The pink buttons allow you, again, through PayPal to donate to our work on a recurring basis. $2, $5, $10 a month in a way that is recurring, repeatable. Set it and forget it. That is one way to do it. Back at the homepage for twist.org or from any page, there is one more link I want you to be aware of, and that is our Patreon link. Click that link and you will head to our Patreon community. In our Patreon community, we regularly post the episodes. We have a special patron RSS feed. I post little pictures and notes to people on a regular basis. And you can join at a specific tier of support depending on your financial ability to help us out. So this is a once per month input of cashola. So you can help out at $3.14 a month, $5, $7, $10. At the $10 level, that's when you start getting to be thanked, your name thanked out loud at the end of the show, if that's something you're interested in. Going on up, you can get some hand-drawn art from Blair. There are lots of fun rewards for helping out TWIS. Look at that on the page right there. Exclusive, patron-only lens or the tier. Yes, there's special stuff only for patrons if you help us out over on Patreon. That's right, Patreon, helping us go on and on and on and on. And so these are the ways you can help us out. Subscribe, buy our merchandise, and donate and support. And finally, tell friends about TWIS. Share us on social media, and tell people who you think you might enjoy it at the next cocktail party you're at. Oh, you like science? You might like TWIS. And then get them to subscribe on their phone right then while you're talking to them. Do it. Everyone, you are how we are able to keep this show going. We couldn't do it without you. Thank you for your support. That is the wrong dial.
Can't explain things you've heard with more than intuition. A lot of reason shows to wait go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scattered plot.
And we are back with more This Week in Science.
Yes, we are. And it is time now for one of our favorite segments of the show. This week in What Has Science Done For Me Lately? This week, let's see if I can actually get the entire message on my screen. Jacqueline Boyster writes in, and Jacqueline says, "Hello, Dr. Kiki, Justin, and Blair. I have been all over the place on trying to decide what to write here. Science has impacted my life and the world in a million ways every day. The more I thought about it, the more this task became daunting. However, there is one very specific thing that science has done for me that I would be remiss to not talk about. In February 2014, I had finally finished school with my very hard-won BS in environmental science. The world was my oyster, and I was going to protect it with everything I had in my arsenal. I was on top of the world." April rolled around, and I went into the emergency room with a severe headache and eye pain. I mean, this sucker was painful. After many MRIs and 2 days of the most awful anxiety I had ever felt, I was diagnosed with multiple sclerosis. From that day on, my healthy lifestyle took a severe nosedive. I was depressed, in pain, my balance was absolutely awful, and I even started to lose normal function in my fingers for a while. My life as I knew it was irreversibly altered. Now, this isn't a sad story. While yes, I have had my ups and downs, and it has been a learning process, I have had to relearn my body and its capabilities. I still struggle sometimes, but the great thing is I have science in my corner. Through science, we are learning even more about this. 10 years from now, we may have even better ways to treat MS. 20 years from now, we may even have a cure. So what has science done for me lately? It has given me hope. Just like it gives millions of people hope for a future where they can feel like the world is once again their oyster.
Oh, thank you so much for sharing this.
Hope.
Jacqueline, thank you so much. Your story is touching, and I do wish you the best in your struggles and also your successes. And I am so glad that you do have science on your corner. I mean, the advances in our understanding of in multiple sclerosis, the treatments that work. We have come light years, and we will get there, and you'll see it happen.
And I can't reiterate it. I feel like I say this now every week. Now is the best time to have any disease as compared to any other point in history.
And it will get even better.
Well, and the thing too is like when we look, like, doing this show gives a tremendous amount of perspective on a lot of things, but especially when it comes to the treatment of medical conditions that have so long plagued mankind in various ways. We are really on the precipice of radical change across a broad array of tools to tackle these things now. And every week as we do this show, there's more and more of that arsenal that's working for you. So
Absolutely. So everyone, remember that we need you to tell your stories of how science is helping you. What has it done for you lately? Whether it is something simple, whether it is something complex, whether it is uplifting, or even something a little sad. We'd love to hear your stories. We really love hearing them. So Leave us a message on our Facebook page. That's facebook.com/thisweekinscience if you still go there. Or you can email me, Kirsten, KIRSTEN, at thisweekinscience.com. And we want to keep filling this segment of the show with something from you moving forward every week. So we need you to keep writing in. Let's hear more stories. Jacqueline, thank you so much. Really do appreciate it. Justin, you're going to tell me how my brain is going to be preserved forever and I'm going to be smart and have no neurodegeneration and it's going to be wonderful, right? Or were you going to tell me about beer?
No, that was the story, but you just ruined it. You gave away the ending.
We win.
So, yeah. So actually, this ties right into what we were just talking about, though. This is potentially the cure to Alzheimer's. This is potentially the way to maybe reverse, but more even likely to prevent it from setting in the onset in the first place. So we have these things around our brain that weren't supposed to be there, right? Let's start at the beginning here. Aging vessels connecting the brain and the immune system play a critical role in both Alzheimer's disease and the decline of cognitive ability that comes with time. This new research is revealing that by improving function of the lymphatic vessels, this is a part of our immune system surrounding the brain that up until pretty recently didn't think that was a thing. That was not part of the brain, wasn't part of the immune system. It wasn't in a textbook. Wasn't there. But it is now. And it's been there for almost 3 years. Or about 3 years. Right? Like before then, this wasn't even there. But by improving the function of these lymphatic vessels that 4 years ago didn't exist, scientists at the University of Virginia School of Medicine have dramatically enhanced older mice's ability to learn and improved their memories. And they say, of course, this could lead to a path. It's not the path, but it could lead to the path to treat or even prevent Alzheimer's disease, age-related memory loss, and a host of other neurodegenerative diseases. And the research is from the lab of neuroscientist Jonathan Kipnis, whose team was the one that made the 2015 discovery that the lymphatic vessels existed in the first place. So they didn't just say, hey, huge discovery, we're done. No, they kept going down the road that leads to the path, right? And actually, if you listen to Kipnis, he says this is more important, actually, than just the initial finding. Quotey voice, when you take naturally aging mice and you make them learn and remember better, that is really exciting. If we can make old mice learn better, that tells me that something can be done. And I'm actually very optimistic that one day we could live to very, very, very old age and not develop Alzheimer's. Because it turns out, leaving Cody voice, it turns out the lymphatic vessels, they are part of our brain's ability to take out the trash, right?
Waste removal system.
Kipnis, who is the chairman of UVA's Department of Neuroscience and the director of its Center for Brain Immunology, and his colleagues were able to use a compound to improve the flow of waste from the brain to these lymph nodes in the neck of aged mice. The vessels became larger, drained better, and that had a direct effect on the mice's ability to learn and remember. Here is the first time we can actually enhance cognitive ability in an old mouse by targeting the lymphatic vasculature around the brain, says Kipnis. By itself, it's super, super exciting. But then we said, wait a second. That's the case. What's happening in Alzheimer's? The researchers determined that obstructing the vessels in mice worsens the accumulation of harmful amyloid plaques in the brain. Amyloid plaques in the brain are associated with
Alzheimer's,
right? So now you have this thing. It's like, oh, there's a correlation between Alzheimer's and amyloid plaques, but it's associated with, and it seems to run with. It's always there but we don't know why. Well, they found one way now in which you could cause it to happen, which could mean that this lymphatic system is strongly correlated at this point, but could be direct causal mechanism later if the research follows this path and discovers that. But this may help explain the buildup of such plaques in people. And it goes on quotey voice here of, this is quotey voice of Dr. Sandro de Mesquita. In human Alzheimer's disease, 98% of cases are not familial, so it's really a matter of what is affected by aging that gives rise to this disease. As we did in mice, it will be interesting to try and figure out what specific changes are happening in the old brained lymphatic system in humans, so we can develop specific approaches to treat age-related sicknesses. Kipnis noted that impairing vessels in mice had fascinating consequences. What is really interesting is that the worsening pathology, with the worsening pathology, it actually looks very similar to what we see in human samples in terms of all this aggregation of amyloid protein in the brain and meninges. By impairing lymphatic function, we made the mouse model more similar to the human pathology. So there's another piece of the arsenal right there. It's not only, hey, here's this correlation, this could be the mechanism. They also, at the same time, may have created a better model for Alzheimer's in mice.
Right, something that more accurately represents it in the neurological system.
Yes, so what's also then fascinating is that then you have a model that might be more accurately able to be utilized to test things that in the past may have been promising, but we don't have a really good way of testing this, right? So other treatments that have been proposed or showed some promise but lacked a good model through which to test or prove or see what the actual result would be. Plus now that we know there's a lymphatic system that could be possibly involved with this, it also means that by making sure that the lymphatic system portion is intact and is functioning properly, that you can also then narrow down what other things are involved as causes. And as they apply, then again, other treatments that may have shown promise in some instances, but across whatever studies were done, didn't ultimately show to have a high enough percentage of efficacy. That efficacy may be related to the fact that there was an impairment of a lymphatic system, and that was what was in the way. So there's this is a one
Hey,
study.
researchers, go back and do everything over again. No.
It kind of is, though. It kind of, or that proposal that you had written up that never got anywhere because there was no way to test it, or that the couple tests that you did, were blocked or had an obstacle that was unseen at the time. So this is one study that is a catalyst of possibilities that flow from behind it. So.
And even beyond, I mean, it's obviously implicated in Alzheimer's, but even beyond Alzheimer's and other neurodegenerative disorders that come with aging or that come with a deteriorating lymphatic system. Like, what else could it be affecting in the aging of the brain?
Yeah.
I mean, and if it is affecting just regular cellular function because waste is not being removed appropriately, there are a lot of cognitive effects that could just be having because of the effects on the neurons themselves of, not sitting in the cleanest environment anymore.
Yeah. Kipnis, final comment here. It may be difficult to reverse Alzheimer's, but maybe we could be able to maintain a very high functionality of this lymphatic vasculature to delay its onset to a very old age. And he says, I honestly believe down the road we can see the real result of extending the point at which Alzheimer's would set in to beyond the lifespan of a human being.
Yes.
Which means basically a form of immunity from this disease.
Until people end up living longer, and then you're in trouble again.
Well, then you just got to keep.
Yeah.
Yeah. Well, but this is like.
It's a give and take. It's a give and take.
Yeah,
You know.
The I'm looking for a word that says is it a watershed moment? Is that a thing? But the discovery of this 3-4 years ago,
it's set up for
coupled
it's
with
many
this
discoveries to
next,
come. Yeah.
you know, the same lab turning around and making that next level discovery, and now opening up an entire approach to a host of diseases that, for all the work and money and time that we've put into them, we were missing one of obviously the key components of the system. It's sort of like trying to address blood pressure without understanding anything about cholesterol or arteries, right? Like oh, so the like it's adding. Oh, but it's fantastic, and so this is definitely something I think we'll be excited to track going on to the future.
Yeah. I mean, one question is just, what with the lymphatic system, like I said, what else is it affecting? I mean, is this involved in migraines? Is this involved in other cellular function? Like, what is happening? I want to know.
Yeah.
And I want to live forever.
Me too.
I want my brain to support me. That's right. So keep working on that, scientists. Let's make it happen. Let's make it happen. Okay. Let's talk about finding a fungus.
Among Us?
Finding a fungus among us. That's right. This story, actually, I came across this story last week on Twitter as it was told by the main scientist from the lab, Michael Eisen, as a tweet thread. And I was just like going through Twitter, and all of a sudden this amazing story unfolded in front of me, and it was fantastic. It's been written up very nicely by Ed Yong, as he is wont to do over at The Atlantic. So the story, Michael Eisen and his team at UC Berkeley have been trying to isolate a fungus in Drosophila. Okay. And so around 2015, Carolyn Elia started leaving fruit out in the backyard to attract wild Drosophila because they couldn't seem to get this fungus to work the right way, because the fungus, I guess this fungus is one of those wonderful behavior controlling fungi where some insects from the wild, these fruit flies, they arrive and died at the plate, their wings erect, and a fungus sprouting out through their backs. This fungus is called Entomophthora muscae, and the name means destroyer of insects.
Uh-huh.
Yeah, it's a great name for a fungus that kills these flies. And it has been killing flies. It's been killing flies in the wild for a long time. Basically, when you find these wild flies, you find the fungus, and that's pretty much the way it works. So the fungal spores end up landing on a fly, growing into the body, starting to eat it alive. And then when the fly is about to die, it makes the fly climb to a really high perch and extend its mouth parts as if there were food to eat, and then get stuck to the perch because the fungus produces a glue-like substance that attaches it to that high point with its mouth out, like grabbing onto the perch. Anyway, the wings raise up and this fly is rictus mortis, and then it dies. But hopefully the spore then is able to, the fungus is then able to grow out of the back and throw more spores around, right? And continue the process ad infinitum. This is a really interesting parasite fungus, mind controlling, right? It's fascinating stuff. And so they haven't been able to get this behavior or really this aspect of the fungus to work in lab Drosophila. And they've been trying to figure out why they can't. And so Michael Eisen in his lab, they were trying to find the microbes that were potentially manipulating fruit fly behavior, Drosophila being, a lab animal for years and years. And so if he could find it, then he could figure out how the neuroscience of the brain control is working, and that could be applied, and the genetics of it, and be able to apply that to all sorts of other things. Well,
So
anyway,
there's something different. There's something different in the lab environment with these flies, right? Than is being experienced by these flies in the wild.
Right. What could it be? And so Carolyn was trying to get wild flies. And so she was leaving fruit out to get these wild flies and to isolate this fungus. So she became the first person to isolate the fungus and get it to infect lab-reared Drosophila. And the researchers started studying it, sequenced the fungus's RNA, and to look at its transcriptome, like what proteins were getting or genes were getting turned on and what proteins were getting made. And they found that 10% of the sequences, these genetic sequences, matched a virus. And this virus had been discovered a few years back by some British researchers that found the RNA in wild Drosophila. So there's this viral DNA. And then Michael Eisen was just like, it's probably not a virus. It's probably a retrovirus or something where just viral DNA. We have viral DNA in our genome. It's probably just gotten stuck in there somewhere. And so he just didn't think much about it and he let it go. He's like, boop, that's a weird quirk. Not interesting. I'm doing more interesting things, right? So they completed sequencing the fungus's genome.
Not be preoccupied by a virus.
I will not be preoccupied by this virus, but they kept working on the fungus. They went beyond the looking at the RNA. They actually started looking at the genome, got all of its genes. There were no viral genes in the DNA
of the fungi,
of the fungus. So they're like, then what we had was a virus either with the fungus. Yeah, there was a virus in the fungus that was in the fly. So we've got like
perhaps she'll die.
Perhaps she'll die. Perhaps the fly will die. Exactly. And they determined that it was a very little known family. It's from a family of viruses called iflaviviruses, and they infect insects. So this entomophthovirus, which they renamed, they decided the British team didn't have the right name for the virus because they're like, it's not just, this virus isn't just infecting the flies, it's infecting the fungus. So we have to change the name. So now it's a renamed virus, and entomphthovirus, it infects a fungus that infects insects. So it has jumped kingdoms.
Yeah.
These viruses normally just infect insects, but this virus doesn't do that because they didn't find the virus. They couldn't get the virus to infect the insects. It only infects the fungus that infects the insects.
Okay, right.
And so what they've been doing is like looking at this, and pretty much they've determined whenever they find the fungus, they find the virus, and they think that the fungus can lose the virus, but when it loses the virus, it loses the ability to mind control the flies.
Wow.
So the virus infected a fungus, which allows the fungus to brain control the flies.
So is that a symbiotic relationship? Potentially.
And so this is a really interesting question as to what the virus does for the fungus, right? Or what the fungus does for the virus.
What the fungus does for the virus, right? Because the virus is allowing the fungus to mind control.
No. See,
so the other way is
wait a second though. Wait a second. Maybe you're assuming that that's what the fungus wants to do.
Right.
But what if the virus is mind controlling the fungus so that it can then use the fungus to mind control the fly? This is all the virus's idea. Like, it's all right. Like, we don't know. Like, no.
Virus.
But the thing is, now we have to know, right? We have to know if the zombie ant fungus has a viral constituent to it.
Right. So that's a very interesting question. And the flaviviruses, these viruses that infect insects directly control this family of viruses controls insect behavior. So if you inject it into ladybugs, like parasitic wasps inject the virus into ladybugs, and the ladybugs become bodyguards for wasp eggs. There's another that makes honeybees more aggressive behaviorally. So the family that this comes from, they do control insects, but this is the virus controlling the fungus, controlling the fly.
It's using tools. This is the first evidence of tool usage in viruses.
Viruses, I love that.
This is amazing.
Yes.
Yeah. So it's an amazing story. They are going to do some tests to determine if the fungus is releasing chemicals to influence the flies with or without the virus. They're going to do, because they have to confirm this hypothesis 100%. But yeah, they do believe the hypothesis supports an idea that this virus is the source of all the mind control, not the fungus itself, actually, as was thought previously.
I'm calling this tool use by virus. I think
I like it. I like that a lot.
But no, I have to know now, like if the fungal zombie ants and the crickets and the rest of these, if there's this viral component, because then that's wow. Then we source all of that activity back to a virus family that would already be looking for ways or finding ways, or however you would propose that it's design or evolved to, or whatever it's however much anthropomorphization you can do with a virus, but that they're conferring abilities.
Right. And so the new methodologies, that they have used, genetic sequencing, transcriptome sequencing, RNA sequencing, looking at these various aspects of the different organisms to put this puzzle together. It's stuff that's being more and more widely used. And this and see if I can say the name, entompthovirus, not the entompthovirus, but the fungus itself that was discovered, this entomoptera. There we go. Entomoptera. It was discovered in 1855. So it's been around and well known for over 150 years. However, this is just being discovered now, thanks to technology.
And what a fantastic, I don't know if this discovery comes about this quickly in the lab. If the virus was in had gotten into the fungus in the lab, because then the fungus has some retrovirus and that's whatever, and why look, it's the fact that was separated from the wild strain that actually
a very special situation.
Mm-hmm.
Yeah,
it's also a good reminder of that whole, I don't know, it has some virus DNA. Who knows? Whatever. Moving on. It's like, wait a second. Let's do our due diligence here.
Yep. Keep looking, even if you're like, this is what science is about.
That's what science is about. Confirming.
Continuing to follow the breadcrumbs
and again,
confirming.
Is it junk? Confirm it's junk before you move forward.
Yeah. I think we have massively underestimated, and science is now starting to point us in the direction of how much influence viruses have on evolution. The ability to generate DNA, to create new DNA from whole cloth, and also to spread this into the environment and act upon other biological life. It's an important characteristic of what life is, these viruses. And I don't think we would be here at all without them.
Yeah. Well, I have another story about fungi / yeast. But before we get into that, I think you have something that is related to yeasts in a way.
It's hard to tell who you're looking at because we both have the same camera, but mine doesn't really have to do with the yeasty part. This is University of Adelaide. Researchers have uncovered fundamental new information about the malting characteristics of barley grains. This is kind of an interesting idea. So they found a link between enzymes involved in malt production for brewing and a specific tissue layer within barley grain. So when you make beer, you've got the barley grain and it gets cracked and then you boil it and it's releasing enzymes. And these enzymes are what convert the starches into the sugars, which later the yeast will eat and convert into alcohol, which gives beer an important characteristic outside of its flavorings. What they kind of zeroed in on is this layer of tissue in the barley grain called the alerone, which is now they've correlated very specifically to what is considered a good barley in brewing, whether that's a craft brewing or what have you. And the thicker this one layer in the barley grain is, the more of this specific enzyme is produced and the quicker and more efficient it is at converting these complex sugars into free sugars, which are then turned into alcohol by the yeast in fermentation. Something that was totally unknown. There's enzymes in there. You try these different barleys, this one did a really good job, that one didn't. But it's now down to this very specific layer. So basically what it's going to allow them to do is to maybe zero in on the genes responsible for creating this layer, do more do cultivation and breeding programs, and come up with a better barley for beer production. Yeah. And it's beta amylase. I mean, it seems like you could just add enzymes.
You probably could just add enzymes. Just use the regular old barley that hasn't been mucked around with. But I prefer to use the science barley.
Yeah, science barley. Is that your new beer brand?
That's right. This beer is science barley.
Beer by science.
Science barley brewing.
Made with science, love, and yeast. And malt, I guess. But anyway,
this yeasty fungusy thing you're talking
about. I'm not going to talk about malt. I'm not talking about barley. I'm talking about yeast. Yes. Now, many people we use yeast in agricultural and also in food purposes, fermentation, making beer. However, there is a particular yeast species that causes a lot of people infectious problems. Candida is becoming more and more prevalent as an infection these days for some unknown reason. We don't really understand that very well. However, this is the tale of two yeast species. The food industry using one species for sourdough bread, bioethanol, beers, and the other.
That's Saccharomyces cerevisiae.
Yeah, well, another species being used, not being used, being terrible and causing yeast infections. But according to a PLOS pathogens report, in which the researchers did genetic analyses of these different species of yeast, they're not different species. They're the same species, but just at different parts of their life cycle.
Huh?
Yeah. One is sexually reproducing, and the other is asexually reproducing, and that seems to be part of the major difference. So for decades, this yeast, Candida krusei, was discovered in 1980. The asexual form, thought of the asexual form of a known sexual species, Pichia kudryavzevii, and the Pichia is safe according to the US FDA, and it's used to make a bunch of fermented items and craft beers, bioethanol, as I've mentioned. However, scientists continue to consider them different species. According to one of a professor, co-author, Kenneth Wolfe, who's a professor of genomic evolution at University College Dublin in Ireland, Candida is simply an old name for any yeast species that doesn't have a sexual cycle or where you can't observe a sexual cycle. Historically, in the days before genetic sequencing, scientists used a number of morphological and biochemical tests to determine whether a species liked a specific temperature, sugar, and biochemical environment to classify them into a specific genus. And so, according to Wolfe, Candida is this kind of like a huge trash can of a species which people traditionally weren't able to identify.
Oh, man.
And so it was this morphological, behavioral differences that led to the naming of various yeast species under the Candida moniker. And so there was this idea that was postulated, I said, by in the 1980s that they were separate species. It was actually put to the test. Researchers, Wolfe and the people he worked with, they compared the genomes of the two species and they expected they'd find a close relationship. However, they found 99.6% sequence identity between the two strain types. And so it's clear that the clinical and environmental strains are the same. So naming is important. I often wonder, it's like, what's in a name? You know,
Yeah.
a rose by any other name would smell as sweet, according to Shakespeare, right? Well, this is actually very important to our understanding of the infectious nature of different species, of what species we're actually working with, their ecology, how they exist with us and around us, and even on their own without us. So the naming here is of a lot of importance, and it highlights the lack of communication and crosstalk between medical and industry professionals, the professionals within the food industry, who've been working on the same organism the whole time, but calling it different things. And so increasing communication can help to reduce misconceptions and potentially lead research in the right direction. So there are going to be tons of stories, tons of research reports out there from the food industry or from the bioethanol industry calling their bacterial species by one name when it really should be another. A bacteria by any other name might not be so infectious. The yeasty difference of a name. Yes. Hey, anyone else have a story?
I have a quick story about mangabees, one of my favorite kinds of monkeys, and a study looking at them and their affinity for nuts. They really like to eat nuts, but the problem is this study from the University of Amsterdam looking at videos from camera traps. They wanted to see how these monkeys were eating nuts and kind of the tactics around that, because they are not good at cracking nuts. So how can they eat them? These nuts are too hard. They cannot open them using their teeth. How can they eat these nuts if they can't get inside of them? Well, there are other animals in the forests where they live that eat those nuts. Animals like chimpanzees and squirrels and hogs, red river hogs. So all of these animals do crack open these nuts, and they wanted to see how the mangabees took advantage of those animals' habits in order to get the treats that they desired. And what they found was that mangabees were present more often at what they called nut cracking sites after chimpanzees and red river hogs had cracked some nuts, suggesting that they were attracted to, quote, nut cracking events. But the white-breasted guinea fowl in the area, the helmeted guinea fowl, some other squirrel species, they were not present more often at nut cracking sites or after nut cracking events, which means mangabees, but not other species, were actually approaching nut cracking chimps and hogs to profit from their nut cracking skills. Why is that weird? Chimpanzees sometimes eat mangabees.
Oh, yeah. Okay.
Yes. So what they found from these camera traps is that at some moments the monkeys seemed to be scared of the predators, while at others they would approach the chimpanzees within a couple meters to profit from the chimps' tool-using skills. They actually crushed and cracked open the nuts using stones. So it led these researchers to believe that potentially they could anticipate the chimpanzees' moods. So this is interspecific communication, whether it's intentional or not. These mangabees are most likely able to read the mood of the chimps, the body language, all that kind of stuff, to figure out if the chimps are feeling carnivorous in that moment or not. And this is interesting because this is something we haven't studied a whole lot, is not just one species' affinity to fall around another for food to acquire free food, but this is also reading the social situation, right? And that they actually think that this same type of decision making might have been present, this is kind of a stretch, might have been present in our early human ancestors in order to obtain free food that might have been hard to acquire otherwise from dangerous predators we may have scavenged off of leftover food. So studying monkey behavior might give us insight into some of our own decision-making skills and the origin there. But also the researchers want to tack on here that this is on the Ivory Coast, and that when we lose these areas, so the forests where these animals are from are disappearing pretty quickly. And so just at the end there, they want to say, when we lose these forests, we're not only losing space for these animals and space where we can do studies on these animals, but we are losing keys to our own evolutionary history. So there you go.
Indeed. Absolutely.
Yeah.
We lose out. We lose the forests. We absolutely lose out. But honestly, are those mangabees just really hungry?
I think they just
Maybe it just has to do with their hunger level.
Maybe. That is a great point. And that's something that they would have to have to check it with maybe checking the feces nearby. If they could, after a camera trap, if they could test the feces for certain hormones and try to see something like that to see how hungry they are. I think that's a really good point. But I think also this isn't their main point of food. This is like a treat for them. So my guess is if they were really hungry, they could eat something else, some fruit, something like that. But
That's right.
they just really want those salty nuts. And so they'll follow the chimps and the Red River hogs to get them. They'll fare being eaten by a chimp, which is just terrifying to me, in order to get some cracked nuts. There we go.
Go nuts. Go nuts, mangabees. Go nuts.
Yeah. Go nuts.
Justin, do you have a last story?
Yeah. So I'm looking over the story from the Journal of Sex Research. Okay. And
as you do.
As one does on a Wednesday.
One does on a slow Wednesday night. What's sort of interesting about this is, well, they surveyed, this is a British survey, but they did a survey that found something interesting, where they asked men and women a number of questions, but one of the specific was number of sexual partners they've had in their lifetime. Men reported an average of 14 lifetime partners. Women reported 7 lifetime partners.
This is pretty par for the course. I feel like every time we've had this kind of a survey come up, this is usually about where the results land.
So when they eliminated the top 1%, right now we've got about 7 partners difference between the men and the women. when they got rid of the top 1% who were reporting 110 partners lifetime for men and 50 partners for women, it actually brought down the whole. They got a whole lot closer. It's about 5.5 now. Instead of 7 different from men to women, it's now only 5.5 different. If you get rid of just the 1% who are the what would you call
They're
them?
the extremes.
The extremes. But even in the
They're
extremes,
not-
the men were at 110 and the women were less than half that.
Yeah.
Okay.
So either this is exaggeration, and so they should be left out, or they're the only ones telling the truth. No.
Do you really keep track of 110?
Right?
Are you really like, yep, that was 109 and that's 110? Are you
So-
really keeping track that high?
So they did go into some detail about the idea that women were more often counting. Of course, they had smaller numbers overall, where men were more estimating.
And does one lead to the other? If you are likely to be counting, will you be
Right.
more conservative with your decisions?
So they also did some interesting things. They got rid of the pay for sex thing, which actually brought the number down almost inconsequentially. 5.5 to like 5.1. Like, didn't make a big difference. They also tried to eliminate, like, how often do you have sex with somebody from a different country? Because maybe that's why the ratio was off. Maybe men were just having affairs with people who were from abroad, and women only wanted to stay at home. And that didn't really make the difference. So they then go into this whole thing about it having to do with figuring out how to get good surveys, because men are obviously overestimating that. But
Yeah,
one of
how
the
do we get real data here.
but one of the interesting things is this. Well, this assumes, first of all, it assumes that men are liars on the subject. They also did sort of hint at, like, maybe women are less likely to report because there's something about the being rough trade or something that a woman might not want to be. So that could also be part of it. But if you take all the honest answers, one of the things I think that I found that they have failed to do is account for heterosexuality versus homosexuality. Now, they say in the report of the story that they didn't exclude based on gender preference, because some people who are of different primary focus also have had the experiences lifetime the other direction. But if you look over the survey doesn't seem to be different for men versus women. It's how many men/women have you So
It's just partners. It's just asking partners. It's not.
Right.
It's. I don't think you necessarily do need to. Characterize it in that way.
Then what you're illustrating is that men, who I think we could agree possibly tend to be a little bit more goal oriented when it comes to dating.
Yeah.
Just I don't know that I'm out on a limb with this. I think this is solid anecdotal. If there's two men who are both goal oriented, that should put the number on that side of the tally much higher, which is where it is. So it actually kind of just could be like, that's makes sense. But they seem so puzzled by it that they're like focusing on, how are they doing their math?
Right.
How could this be possible that their math is so different than what the women report? Because there's only so many women over here. How could we possibly have more men on this side who are having, like, it just does not occur that because
interesting
there could be more activity over there. There could be a lot more. There could be more stuff going on that side of your tally than this side. So I guess I don't know if you can take a takeaway from this, but if sexual partners and having a long list of them as notches on a belt is something that you're shooting for in life, you're better off as a gay man. than anything else. I think is like if you really want bragging rights, gay men have all the bragging rights. Nobody else apparently can touch what they're doing because they've moved the statistic.
Yeah.
Like a full they're almost doubling in some instances what the other side is able with the other preference side is able to. Anyway,
110 still bugs me. Like, how do you come up with that number? 110. You're like,
right?
Definitely more than 100, but less than 120.
But what's also interesting,
Even no less than 120. How at a certain point, like
it's just a lot.
but wait, what's also
wouldn't it be? I don't know.
What if there's a group of people who aren't having any sex?
I am sure there absolutely is.
Yeah.
So with that,
Mm-hmm.
but if that's the case, that means women's selectivity could be increasing the male count on the other side. It could be like the guys who are polled who are like, oh, I'll tell you about my sexual encounters, and I've had a lot more than the women on your side of it. But then there might be this huge population of uncounted men who are like, oh, it was that one time, but that was it, right? Like this could be the thing. Women are selecting the same guys over and over again, right? Like that could also be influencing these numbers.
Yes, that's true.
Yeah, so I guess what it does go to show is that statistics really are just numbers, and there is more beneath them that needs to be elucidated, and it's hard to find out. You sometimes have to dig to understand what's actually going on.
Statistics are a fickle mistress.
A fickle mistress. Yes. Another fickle mistress is Mother Nature. Mother Nature, with its increasing frequency of very strong hurricanes in the Caribbean, we're looking potentially at a story of anal lizard adaptation and potential evolution as a result of really strong hurricanes. So, researchers at Harvard University traveled to Turks and Caicos in the Caribbean to study local anal lizards, and they were looking just to see the native wildlife, and they wanted to know how lizards might evolve once rodents were off the island, because conservationists were trying to exterminate rats, and that might give the animals more food sources. And so that's really what they were looking at. They were going down there, but then boom, Hurricane Irma battering the island, and then Hurricane Maria battering the island, and more hurricanes. It was pretty intense. They didn't really they'd seen the animals before the hurricane struck very briefly and done a few measurements ahead of time, and then they went back after the hurricanes. Were like, oh, maybe we'll see some lizards can grab onto things pretty well, so maybe they didn't get thrown into the ocean by the hurricane force winds. They didn't think that there would be a really big population, but when they got there, they found that the lizards had changed. Yes. It wasn't a very dramatic change. Their toe pads on their front, or their toe pads in general were 6 to 9% bigger than their pre-hurricane lizard compatriots. Their front legs were longer by 2%. And when they looked at the hind legs, they found the hind legs were 6% shorter. They found the hind legs of these lizards were 6% shorter.
So is this weather selection for like lizards who can hang on in a hurricane?
That is exactly what this is. They took animals on. I just have to show you. I have to show you a picture. Blair's showing you lovely pictures of these lizards, but I have to show you a picture of their experimental setup to test what was going on with the hind legs. They couldn't really determine. They're like, why are the hind legs getting shorter?
Not what you need. You gotta hang on with you're hanging on with the front. Your back ones are just dangling in the
That's right.
Wind.
Yep. So the lizards, they put them on sticks, and then they took leaf blowers and aimed them at the lizards. and looked at what happened. And they found that the hind legs don't hang on as well, just as you thought. The front legs are the grabbers, and shorter legs and bigger toe pads help with their grippiness. The hind legs, the longer they are, the better sails they make. That's more leg area
Oh no, which is
to be
bad if you're
lifted
trying
up
to
by the
be-
wind.
Yeah.
Yeah, and so they determined that shorter legs are they're less good as sails, and so the lizards are better able to hang on with the shorter legs.
What do you
Yeah.
do for research, Belinda? Oh, I point leaf blowers at lizards.
Well, this gives me confidence in a hurricane, though, because I've got good sized hands, long arms, and short legs, so I'll be fine.
You're perfect.
Yeah.
You're in a knoll.
Fine
You're-
for a hurricane.
You're just right for hurricane weather. That's right. So while people might say that, oh, this is evolution in action, it is part of evolution. This is kind of the force of natural selection in action, environmental forces leading to some individuals surviving better than others. And we will see how their offspring do, which ones succeed to pass on their genes year to year. Evolution is more complex than simply, one stimulus in and a result out. But so far, short-legged animals hang on better. So
Where
say
did the other ones
we
go?
all.
Like how? Are they all at sea?
Yeah.
They got blown into the ocean. That's right.
Are there any more islands in that direction, or that was the last island? I don't know.
There are lots of islands through there, but yeah, who knows? That's right. Maybe another one of the islands, like the driftwood and lizards, are going to be showing up.
Further north, there's this island with all these short front-legged, long rear-legged lizards that are just
That's
totally
right.
taking over the.
Justin, evolution, like statistics, is a fickle mistress.
All right, everyone. I think we have come to the end of our show. Have we done it?
Yeah.
Oh yeah, I think so.
We have done it once again. I want to say thank you to everyone who was watching live. Those of you in our chat room, thank you. Thank you for your great conversations over on YouTube and in our main chat room. Also, thank you to those of you who are listening right now. If you're listening in your car, I'm glad we were able to keep you company wherever you are. Thank you to the people who helped to keep this podcast going. Identity 4, thank you for recording the show. And Fada, thank you for helping in our chat room over on YouTube and also with social media and with the show notes in the description on YouTube. He spends a lot of time putting in specific time codes where you can easily find stories directly through the description. Does a lot of work there. So thank you so much.
Yeah, and if you're watching on the YouTube and you've liked what you've seen, you just fell in here, you can blow us, click on the subscribe link, and then you would be able to get updates whenever there's a new show.
I'm supposed to point down now. Subscribe. Okay, and I want to thank our Patreon donors. Thank you. 2, Paul Disney, G. Burton Lattimore, Richard Onimus, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Andy Gross, Steve DeBell, Alex Wilson, Tony Steele, Ed Dyer, Craig Landon, John Ratnaswamy, Mark Mazzaros, Jack Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Kyle Washington, Eric Knapp, Richard Brian Condren, Aiden Jeffs, Kevin Reardon, Dave Neighbor, Christoph Zuknurek, Noah Zemke, Jim Seabright, Ashish Pants, Jacqueline Boyster, Ulysses Adkins, Shara Chavis, Richard Porter, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim Trapeau, Greg Riley, Sean Lamb, Ben Rothig, Steve Lesseman, Kurt Larson, Robert Aston, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mac Hessenflow, Kevin Parachan, Byron Lee, E.O., Mark, Tyrone Fong, and Keith Corsell. Thank you for all of your support on Patreon. For those of you who might want information about that, you can find it over at twis.org. There's all sorts of information about that kind of stuff. And you can also find information about subscribing. And on next week's show, we will be broadcasting live online once again on my birthday.
Oh, happy birthday!
Yeah, it's my birthday next Wednesday.
It's
So
my birthday too.
yeah, no, it's not, but okay, it's my birthday. Well, it will be my birthday. And I hope everyone comes to enjoy this birthday celebration show. I don't know if there'll be, maybe I'll have a cupcake. I don't know, but it'll be science and it will be fun. So 8 p.m. Pacific time, where we always are, twis.org/live or on our YouTube channel. You can also access that easily by going to twis.org/YouTube.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile type device. You can search for TWIS, the number 4 droid app in the Android marketplace, or simply This Week in Science and anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org. That's TWIS.org, where you can also make comments and start conversations with the hosts or other listeners.
Or you can contact us directly. Email Kirsten at twisminscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Blair's Menagerie, and at Jacksonfly, which is almost all politics right now. So maybe avoid that if you want to avoid that. We love your feedback. If there's a topic you would like us to address or cover, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week for Kiki's birthday, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
Oh, we have reached the end of another show. We have come to the end now, and it is time, it's late, it's 10 to 13 in the evening time, and we have done the science.
You're scaring me.
I'm sorry, did we do the science and I scared you a little bit? It's the spiders! Spiders! The spiders. I like spiders. I don't like them in my face.
Yeah, not in the face.
It's one of those moments. This morning, I went down to turn on the sprinkler in the backyard and ran outside. You know, it's like 6:30 in the morning, and I've just put the coffee on, and I'm running outside, and I'm like, "Bah!" Turn on the water, and I look up, and there's a web, and there's this little garden spider. It's like seriously like 4 inches from my face. Just missed it.
I don't like it.
Good morning, spider.
My 2 least favorite types of spiders, my number 1 with a bullet is the orb weaver, because they do it right at face height.
Yes.
And they
always.
take down their web and put a new one up every night, so it's extra invisible.
That's right. They're not dusty, they're not dirty. They're just pretty and invisible.
Yuck. So those, and then my second.
I love orb webs. I love orb spiders.
Wow, they're so creepy looking. And then my second least favorite is the wolf spider, because they're like quick and they jump. They're real yucky.
Yeah, it is. Everybody talks about fall, you know, being spider season. You have the fake cobwebs that you put out everywhere. But seriously, spring is there's spring, but right now in Portland is massive spider season. It's the spiders everywhere. There's spiders walking all over the place. I've got the walkabout spiders going around my house, and I'm like, what are you doing walking around my house? Okay, go hide. Go away.
Rescued a spider from the lab today.
Yeah, I found a hobo spider.
Down the hallway, and I scooped them up and walked them outside. I love spiders. Spiders are my friends. Anything that eats mosquitoes is my friend. And even if you're just related to a species of spider that eats mosquitoes and you yourself don't partake, you're still on the list. I do not discriminate.
I'm cool with spiders. And for the most part, the spiders here in Portland are pretty harmless, not really anything to be concerned about. But it's just that every once in a while, it's like, you know, like the hobo spiders and the ones that go wandering around. Like they're like, we're just going for a walk and I'm going to be in your shoe. Or, oh, hey, I ended up in your blankets. Or, oops, look at me in the pile of your clothing.
Okay. So apparently there's not as many spiders where I'm at. But the one that does show up is the daddy longlegs.
Oh yeah, they're
fine.
Right. And my daughter was who's now 5, ran into spotted one in the bathroom and was very concerned about it. Like, which is weird because this is like a learned behavior almost, I think, because she's been cool with spiders in the past, but like apparently that got overwritten at some point, and she was like, I can't go into the bathroom. There's a spider. Went look, and I said, okay, well that's a daddy long leg. They don't bite. They're not gonna hurt you. But the thing that he eats is mosquitoes. And my daughter hates mosquitoes probably more than I do.
And she's like, I love the spider.
Yes, her attitude completely swung the opposite direction. And he's like, can we get some more of them? I'm like, no, we'll just let nature work its course. We're gonna let this one live in this corner. We're not gonna take down the little web he's got here in the corner of the bathroom, but we're just gonna let him be there and live there. That'll be fine. But no, I don't think we want to, fill. Now she wants to fill the house with Daddy Longlegs.
Yeah, and then you're like, the cobwebs.
That might be too many. Yeah.
Apparently, hobo spiders are aggressive.
Yeah, they can be.
Ah!
Like what? They cut you off in traffic?
Yeah.
Like, what are you talking about?
They'll jump at you. Like, they stand their ground.
Yeah. Well, yeah.
And they'll bite you. They will bite you.
Man,
and
I think that's what was on my car the other day. Is a hobo spider.
Really? Now that you're looking at it, you're like,
Yeah.
oh.
So I came out. I was just shopping, and I came out to my car, and my car legitimately had like a spider had gone around my entire car. There was a spiral of a web across from the driver's side door over the roof, across the passenger side, under, and then back over my driver's side door. Like, I was
like, hey, spider,
what?
this is not a good meal for you.
This
is
too big
not going to taste
you.
good. Too
well,
big.
To be fair, your car does look a little bit like a dead fly.
Okay, whatever that means. But I did find this rather large brown spider hanging out on the driver's side, but on the back seat window. And I was like, shoot, as soon as I open this driver's side door to get in, it's going to pull on these webs. The spider's going to end up in my car. So I was just out on the street because I had parked just on a street. Like, I grabbed an envelope out of my purse and I was trying to convince the spider to get off of my frickin' car, and he would not get off of my car. And it got to the point where I convinced him to go like farther back on my car, to the point where I felt like I could get in the car real fast. And I was like, okay, as I'm driving, he'll go away. He'll get blown off, right? No, I drove for 15 minutes. When I parked, he was still there. And so that I had to get a little more aggressive. I had to push him with the envelope completely off my car onto the ground before.
I don't even know this neighborhood. I don't know what I'm doing here.
That's not my fault. That's not my fault that he did that.
Oh, goodness.
Oh my God. Yeah. And then, because then I was like, I have to get rid of him, because I was going to my friend's house, and I was like, when I come out, it's going to be dark. I will have forgotten about the spider and I'll end up with a spider in my goddamn face. So I had to kick him off of my car, because I didn't want to end up with a spider in my face when I was trying to get into my car later. I already have spiders living on my car elsewhere. They live in the passenger side mirror. So every time because every time I wash my car, it cleans up all of the webbing, and I've even removed it myself before. And then 2 days later, it's back
in the same spot. They're in there.
So
I'm reading through a PDF document from Oregon's Department of Agriculture about Oregon spiders, facts and fictions. And one of the best sentences here: realistically, although it may not be comforting to realize, our homes are, quote, islands in a sea of spiders. There we go.
I was once told, so when I was a kid, my current situation with spiders is like, I might as well be like, kissing spiders in relation to how I used to be with them. I used to be so freaked out by spiders that if I saw a spider in a room and it didn't get killed or removed, I would not be able to go back into that room, like ever, like for days. At one point, there was a spider over my head during an assembly when I was in like second grade, and I screamed, ran out of the room, made a big deal out of it. I got sent to the principal's office because I freaked out so hard. I could not handle spiders when I was a kid. And so my mom was like trying to do like intense aversion therapy. Like she got me books about spiders from the library, tried to
Spiders
show
are
me
your
pictures
friends.
of spiders up close. She was like, maybe if she knows more about them, she won't be as afraid of them. Did not work out super well for me at the time. But one of the things that she told me that actually just made me lose a couple nights of sleep before it started to sink in was, you're never more than 3 feet away from a spider. You're never more than 3 feet away.
Which can actually be true on an airplane. Actually, this happened when I flew to Portland last
There was
time.
a spider in your plane?
There was a teeny tiny spider that was dangling from a web in front of me, and I was transfixed because as the plane is sort of going up and accelerating, coming down, the spider hanging from this thing is like a little pendulum. He's like starts to move a little closer towards my face, and then the plane adjusts, and it sort of moves back over into this area. But he was some sort of pendulum showing me where gravity was still directed and located on this.
He was your stomaticist on that day.
It was one of these. It was a smallish. The plane that goes from like the Sacramento airport to Portland is like a puddle hopper of a plane. I mean, it's just it's a propeller plane. It's that small. But it was pretty cool. It was a fun passenger. Like the seat next to me, they weren't as interesting as the spider. He was, or she was. I don't know. I don't know how to, especially that small of a spider. Figure out the gender.
I'm looking at pretty white spiders right now. White spiders and yellow spiders and
I'll just look at pictures of jumping spiders all day.
Spiders.
If all spiders were jumping spiders, I'd be fine.
And that's the thing that surprises me about this backstory of spider fears. You do have like a huge affinity for at least one spider on this planet.
Yeah,
to the point where like I would think that's one of your favorite creatures on this earth, and so that all of the rest of its kin are an affront to you. That kind of blows my mind.
It's just I'm working through it. I have to sneeze.
Isn't it?
Oh my god. It's I'm working through it. Like as I learn more about the planet and ecosystems and animals, I couldn't help but start to understand and respect spiders.
So
I have an undying respect for spiders, which is why I don't kill them anymore, which is why if there's one that is nowhere near my bedroom or my face, they are allowed to be where they are. I won't even move them. It's fine. But if they are near my face or they are near where I sleep, they need to be moved.
I'm the same way with cats.
I was house sitting once and I did wake up to a spider scurrying across my face, and I did get a pretty large spider bite on my face that was a nightmare. It my face swelled up, it turned green. It was not great.
You got a spider bite on your face?
Yeah. I covered it with makeup, so you'd never be able to figure it out because I definitely had it during the TWIS years. But
oh my goodness.
Yeah, it was real yucky, and I was like, this is why spiders are not allowed near my face. Because at first I was like, oh, I dreamt it. It was my hair moved over my face or something like that, right? And then the bite showed up the next day. I was like, oh, didn't dream it, turns out.
Nope.
Real yuck town, if you ask me. Real yuck town.
Yeah, that wouldn't be fun. I'm trying to figure out Misumina. What Misumina means. It's very pretty. There's white spiders. There are crab spiders that live around here, Northern California, North America. And they have such a pretty name. They're these cute little white spiders. They're not venomous. They just live in flowers. They're called Misumina vatia. And it's just such a pretty name. Flower spider. I don't know. It's very pretty. Where does it say it? Meaning. Misumina vatia? Nobody wants to tell me what it means. Crab spider. Sounds better than crab spider.
Yeah.
I like spiders. I don't like them near my face either. End of story. Here we go. Oh, Sunny de Oliveira, you're welcome for the stories this week. Everyone over there in YouTube land, thanks for your commenting over there. Absolutely. There in the chat room. Oh my God. Ed from Connecticut says his worst spider experience was taking a relaxing bath. Open eyes to see spider floating towards my face. My scream as I jump out of bath was a very manly scream. Of course. I believe this. Okay, and Fada wants me to go look at the React.js logo. Oh yeah, they just took the Atom logo. It's my shirt is not. I mean, it's geeky. I mean, JavaScript, but it's a science Atom because atoms mean science. See, look in the TWIS logo.
Yeah, we got one.
Atom equals science back there.
Yeah.
Science. It's the modern age.
That and the fact that quarks are just hard to represent.
Yeah, those subatomic particles, they're kind of difficult to put in there. Put all the pieces in. Ah! Spider landed on your head while you were shaving? Bah! Oh my gosh. Noodles says rabbits are a terror to me. Oh my gosh. Is it the ears? The ears are they're very large.
They're not as cuddly as advertised, I will say.
Well, there is
big
a
fangs.
there is a huge difference between a wild and a domestic too.
No, even then, they're not as cuddly as advertised.
I don't know if I buy that.
That's my entire childhood growing up. I loved rabbits. I had about 1 million stuffed animal rabbits. I had all the books about rabbits. I had rabbits on my clothes. I had rabbit bedspreads. I loved rabbits. When I started volunteering at the zoo, there was no animal I was more excited to handle than a rabbit. Like other people are like, I can't wait to handle the snakes. I can't wait to handle the alligator. I can't wait to handle the ferret or the chinchillas or the hedgehogs.
Rabbits at the zoo?
And there were rabbits. And yeah. And so those rabbits were not chill.
Were they wild?
It was a big disappointment though. It was a big disappointment.
Domestic rabbits?
They don't really like to be held. They poop everywhere.
Well, these sound like these zoo rabbits might be different. So had a rabbit for a while. The rabbit used a litter box. The rabbit did not go outside of the box. Only used the box. It was house trained.
I think that's the exception to the rule.
No, it's not. It's a whole thing. You can train a rabbit to be like what a cat would do in that respect.
You can. It's true.
The one thing that I couldn't train the rabbit not to do and ultimately led to its moving to a farm was it anytime it found a wire at rabbit height, it would gnaw through it.
Yeah, of course.
So there goes the internet line. There goes the power cord that was charging my phone. There goes the lamp. What is going on? Yeah. But yeah, it was, cuddly-ish. You can sit on your lap and like pets, and it would just chill there. And then when it didn't want to, it would just be like, oh, I'm going to go hop around. Yeah, see you.
Hop, hop, hop. Hop, hop, hop.
But they are I don't know, maybe we didn't do this story, but it was, it came across in the pantheon of stories that crossed my vision about there being really dramatic brain morphology differences between wild type and domestic rabbits.
Well, that's interesting.
Yeah, which makes me suspect that the rabbits that Blair encountered weren't truly the domestic variety.
They're the
Or-
domestic European rabbit is what we had. And we
Not
also had
well-trained, though, and aggressive.
we had the domestic Norwegian dwarf rabbit also. I mean, they didn't live in a house, so they saw people every day. We had people in with them multiple times a day, every day, but they didn't live in a house. So maybe for rabbits to really be more friendly, they need to be around humans 24 hours a day.
Makes sense.
Possible.
I wonder
But-
if bunnies maybe have a sensitive period like cats do to be humanized.
Maybe.
That makes sense.
Maybe it's something like that.
Okay, what do you say, folks?
Yeah, that's right. You're not feeling well.
I say it's time
You
for
are
you to say goodnight,
sickish.
Blair.
Okay. Good night, Blair. Say good night, Justin.
Good night, Justin.
Good night,
Kiki.
Kiki.
Good night, everyone. Thank you so much for joining us for another episode of This Week in Science. We'll be back next week. Have a wonderful science-y interval in between. Thank you.